Psychiatric genetics has mapped human distress at a scale once thought impossible. Researchers have scanned millions of genomes, identifying molecular variations associated with profound suffering. These genome-wide association studies, or GWAS, rely on a specific starting point. They group people based on clinical diagnoses, like schizophrenia, and search for shared genetic markers. Because these genomic tools are so precise, we have started projecting modern polygenic risk scores backward onto ancient DNA. Allele frequencies shifted under ancient ecological pressures, changes in diet, pathogens, and early social forms, not modern clinical disease. The modern score architecture is real, but the ontological authority we grant the clinical diseases attached to it is far less secure. The problem is a circular logic. Psychiatry uses culturally negotiated DSM criteria to build the genetic cohorts we study. We find genetic markers for those groups, and then cite those markers as biological evidence that the original DSM categories are natural disease entities. If these categories were discrete biological diseases, the genetic data would map to them with precision. A major 2025 study in Nature tested this across 14 psychiatric disorders. The rigid columns dissolved. Researchers found that 5 broad genomic factors explain the vast majority of shared genetic liability across all conditions. Related research in JAMA Network Open shows that disorder-specific scores actually capture broad, transdiagnostic liability to distress, rather than sharply bounded illnesses. These polygenic scores index liability to contemporary institutional groupings, not timeless natural kinds. By using these administrative labels to define the biological input, we have been reading the human genome backward. We should invert the question. Instead of asking which genes cause disorder, we should ask which inherited variations increase a person's sensitivity to their environment. A 2024 molecular psychiatry study highlights differential susceptibility, where the same genetic traits amplify both well-being and emotional problems depending on the context. Just think of a vibrant rainforest plant suddenly move to a sun-scorched desert. Within days, it will begin to fail. The plant is not suffering from a pathological genome, it is experiencing an ecological mismatch. Human behaviors labeled as pathological are often markers of a highly tuned biological system reacting to its surroundings. The genetic signal likely indexes specific sensitivities: salience responsivity, sleep vulnerability, reward attunement, and stress reactivity. In a supportive environment, these same traits can produce high creative output or deep social attunement. Severe mental distress is the breakdown of a highly sensitive organism stripped of its required ecological niche. We must discard the idea of the environment as a single, flat statistical variable. The psychosocial milieu is a layered, historically specific ecology. Studies from Kirkbride and Beyer show that severe psychiatric risk is concentrated in specific urban textures. The social texture of urban life, the noise, the crowding, and the social fragmentation, elevates risk far more than urban residence itself. These urban stressors act directly on shared biological pathways, particularly stress response systems and sleep architecture. The same genetic profile will produce a completely different outcome depending on the neighborhood it inhabits. Psychiatry in an affluent rural setting operates in a fundamentally different ecology than psychiatry in a dense inner city. Analyzing genes without commensurate ecological data produces a statistical hallucination, detached from the lived reality of deterioration. Migration and dispersal disrupt a person's meaning structures and social networks. When combined with racialization and discrimination, the cumulative biological stress compounds until it overloads the system. Material scarcity and housing instability act as everyday environmental toxins. These forces place the body's internal threat detection systems into a state of chronic, inescapable activation. We must consider whether modern Western institutions actively manufacture deterioration. Our schools and workplaces are often rigidly hostile to cognitive nonlinearity and high sensitivity. A modern genetics that claims true ecological validity must actively account for how these rigid institutional arrangements amplify our inherited traits. Institutional language transforms complex personal distress into a process called semiotic capture. A patient's history is progressively narrowed. Complex biographical context is translated into low mood, then depression, and finally treatment-resistant depression. This final label serves the system's need for administrative legibility, not the patient's phenomenological reality. Geneticists recruiting based on these labels are studying the endpoint of an administrative process rather than a natural boundary in human biology. Naming a disorder provides the illusion of an explanation, foreclosing scientific inquiry before it begins. We should demote diagnosis to a provisional shorthand and replace it with multidimensional formulation. In this model, genetics occupies its correct, limited position, forming the predisposing layer of sensitivity. Environment interacts with biology through precipitating losses and perpetuating pressures like deprivation. Most importantly, it accounts for the protective layer, access to nature, social safety, and interpretive freedom. This approach preserves the findings of psychiatric genetics by relocating them within the physical and social realities of human ecology. The ultimate goal is to build a psychiatry subtle enough to understand the precise interaction between a person's inheritance and the world they inhabit.